So welcome everybody. You are now following a Usomi webinar. And first of all, I would like to welcome you, of course. And then for those who don't know me yet, I'm Eric Ranchard. I'm the president of the European Society of Medical Imaging Informatics. I'm a radiologist myself, and I'm working at the ETZ hospital in the south of the Netherlands. And of course, I'm also much interested in everything related to imaging informatics and artificial intelligence. Today we have somebody else in our panel as well. That's Merel Huisman. She's also a radiologist, and she's a senior actually resident at the Meander Hospital in the Netherlands. Merel is, has a special interest for cardiothoracic radiology. And well, as you can see on her LinkedIn profile, she's a really enthusiastic, analytic, but also creative thinker, and that's true. She's a connector, and therefore we are very happy to have her in as a chairman of the young club of the Usomi. We have a young member club, and so if you are interested, you can also join our club for a reduced fee. For all the young club members, you have to be 36 years or younger. So having said this, I'm going to give the word to Merel.
Well, thank you, Eric, for all these compliments, even calling me a radiologist. In reality, I'm indeed a resident, but thank you. So I'm very happy to moderate this webinar and to introduce my former colleague and good friend, Martin Willemink. Martin is a physician with a background in radiology, an epidemiologist, a biomedical engineer, and a clinical scientist. So really he has a very, very strong track record and a good profile to tell you all about AI from all perspectives. Right now he works as an instructor at Stanford University School of Medicine at the Department of Radiology. And his focus is on cardiovascular imaging with a special interest in computed tomography. Apart from this job, he also has co-founded SecMed, a company that focuses on preparing medical imaging data for machine learning. And on top of that, he's also a strong scientist. He has had over 70 publications, and he is right now a junior. It's hard to even pronounce, junior deputy editor of European Radiology. So that's a mouthful. The topic of today is preparing medical imaging data for machine learning. And actually he already published an article in Radiology about this. He did a very good job, I think, in explaining the first steps to get started in the AI space to a broader audience. So democratizing the topic, so to speak. So that's it about the speaker. First, I would like to explain as well that if you have a question, you can write your question in the Q&A box on the bar below, not in the chat, please. And then the speaker will reply to you at the end of the talk. So Martin, the floor is yours.
So sorry for interrupting you, Merel. I just have to tell one more thing. We also have to thank our sponsor, Bracco, for helping us with organizing these webinars. So yes, please, Martin, it's your turn now.
Thank you. Thank you very much, Merel, and thank you very much, Eric, for the nice words and nice introduction. I'm very honored to be here presenting for you and all the attendees. I see the number is still going up, so that's nice, nice to see. Yeah, so today I would like to talk about how to prepare medical imaging data for machine, and I would like to focus on the radiology space. And I would like to tell this from a perspective of a clinician or radiologist that would like to start with getting their feet wet with trying a little bit, experimenting a little bit with machine. So before you can do that, of course, you need to prepare data. You need to have some data to play around with. And that's what I'm hoping to teach you during this lecture. These are my disclosures. Most importantly, I have been a consultant for Arteris, co-founder and stockholder of SecMed, which is a startup company that prepares medical imaging data for machine learning. So during this talk, I am mostly going to walk you through the process of how to prepare medical imaging data for machine learning. And that's going to be the majority of the talk. I'm also going to shortly talk about SegMed, a startup where I'm a co-founder in, and we are focusing on preparing medical imaging data. But let's start about how to do this. So machine learning and deep learning has many potential applications in medical imaging. I like to divide them in three categories: either improving workflows, automatic detection of lesions, and automatic quantification using medical imaging. So an example of workflow improvement is prioritization of work lists. For example, this is a scheme that's presented by Privadello et al., published in 2017 in Radiology. What they did is they evaluated non-contrast CT scans of the head, and they first ran an AI algorithm to detect whether hemorrhage, mass effect, or hydrocephalus was present. If this was present, then the exam was marked as as a critical imaging finding was present. If none of these was present, then another AI algorithm was used to evaluate whether stroke was present or not. And again, if stroke was present, then it was marked as having a critical imaging finding. And if not, then it was marked as having no critical imaging finding. So this is a nice example in how you can prioritize the work list of a radiologist by putting those ones that are tagged as having a critical imaging finding and put those on top of the work list. So this is a typical example of how AI can improve the workflow for a radiologist. Other examples are to triage screening mammograms, to reduce or to eliminate gadolinium-based contrast media for MRI, and to reduce noise for CT radiation dose reduction. These are all examples for workflow improvements. Another application of machine learning and deep learning in medical imaging is automatic lesion detection. And here we can see an example published by Wang et al. Last year in Radiology. And what they did is they automatically evaluated chest x-rays for the presence of pneumothorax. And here you can see an example. This is chest x-ray. It's hard to see on this like stamp-sized image, but anywho, there is a pneumothorax present, and it was confirmed this chest CT scan. And here you can see the algorithm created a heat map that shows where the relevant information was extracted from, and you can see it's exactly where the pneumothorax is present in this patient. So AI can be very assisting radiologists if certain urgent findings, such as a pneumothorax, are present, or findings that are maybe hard to miss. Other examples for automatic lesion detection include detection of intracranial hemorrhage, detection of diseases such as Alzheimer's disease, or detection, maybe a little bit easier, of urinary stones. The third category, automatic quantification. This is a study from David Larson and other Stanford colleagues that was published 2 years ago in Radiology. And what they did was they evaluated the skeletal maturity on pediatric hand radiographs. And this was also fully automatic. Other automatic quantification examples are to do calcium scoring on non-contrast cardiac CT scans, prostate classification on MRI imaging, breast density on mammographies, or ventricle segmentation for cardiac MRI. So as you can see with all these examples, there are lots and lots of opportunities for machine learning and deep learning in medical imaging. However, when we look at our clinical use of AI and machine learning, this is actually very limited right now. So why is this currently so limited? Well, coincidentally, Merel Huisman and Erik Ransgaard are the co-moderators of this session, but actually we did a project together, also with some other people like Tim Leiner and some other international friends. We created a survey in which we interviewed radiologists and residents to evaluate what their attitude was towards AI in the radiology setting. So this was not focused on the key opinion leaders in radiology that where you usually hear, like, this is the opinion, this is what we think as radiologists. This was really focused on radiologists and residents that are working, you know, also in like smaller hospitals, and not the ones that are constantly on a daily basis working with AI and machine learning. And so we focused on what their attitude was towards AI, but one of the topics that we had some questions on was what they thought were the major hurdles for AI implementation. So in total, we interviewed, or more than 1,000 radiologists and residents took part of this survey. And here you can see some of the hurdles that were mentioned by these participants. And most prominently, ethical and legal issues were mentioned, but also lack of knowledge, for example. I want to focus a little bit on these two items. So 39%, so a substantial number of the radiologists and residents mentioned that generalizability of AI software was a hurdle according to them. They also mentioned that high quality image data was only a hurdle, was only mentioned by 15%. So this was a relatively low mentioned hurdle, but I think there's a discrepancy. Because if you want to develop a generalizable AI algorithm, you will need high quality and, most importantly, heterogeneous image data. And a hurdle, I totally agree, the generalizability of the current AI software is a major hurdle, but the cause of that is actually having heterogeneous and high quality image data. So I think that discrepancy here might be caused because the people that filled out the survey are maybe not the people that every day work with this technology. If you ask experts, where is the story, data curation takes about 80% of the work, and the actual model development, so the actual AI algorithm development, is only 20% or less of the time that people spend. And I actually recently saw this one on Twitter, that model deployment, which comes after model development, actually takes even more time. But before we can focus on model deployment, we need to be able to develop algorithms. And before we can do that, we need to actually curate the data. So that is what we're going to focus on in this presentation, because nearly all limitations in the data, all limitations in AI radiology can be attributed to one substantial problem, and that is the lack of available image data for training, validation, and testing of AI algorithms. Within our Stanford cardiovascular imaging group, we actually experienced this ourselves. So we are, one of the major projects that we are working on is complications in patients that have initially uncomplicated Stanford type B aortic dissections. And we first started developing some algorithms and doing some research on our own small cohort. This is an example which was done by Louis Hahn, one of our group members who now works at UCSD as a radiologist. So we looked at CT scans of the aorta. These are all CT patients with type B aortic dissections. And first, the aorta was localized. The second step was to identify the center line based on the aorta and to create multiplanar reformation images, so MPR images. And based on those MPR images with a smaller field of view, a convolutional neural network was used to segment automatically the true lumen and the false lumen. And the last step was to back convert this to actual images. So you can just see your same CT scan as you saw here, but now we have 3D segmentations of the whole aorta, the true lumen and the false lumen in aortic dissection patients. And this is all nice. The idea to do this is to actually be able to automatically extract information that can predict late complications. And the problem that we had was that we actually had a relatively small sample size, and also the data only came from a highly specialized center, only from our own Stanford hospital. So at some point we thought, maybe it would be better if we would actually do some external validation, or maybe we should expand our data set. So more than 3 years ago, we decided to collaborate with multiple hospitals throughout Europe and North America. We are currently having a collaboration with 10 international hospitals. And more than 3 years ago, we started with doing all the paperwork, getting the ethical approvals, getting the legal approvals, getting the data share usage agreements, and those kind of things. And even though we are 3 years in, we still have not all the data in our own core lab database. So yes, it takes a very, very long time to get the data. And some of the data is already in, and we are still doing segmentations to get ground truth information before we can actually develop high quality algorithms. So not only is it taking a long time to get the data, also it takes a very long time to just label the data. And there are many more other issues that we are facing. And yeah, so this shows a real world example that we are facing within our own research group. And the same problems happen in the industry because in industry, generally larger data sets are needed compared to just a research project where you sometimes can work with just smaller sample sizes. So one of the problems in data is that data is stored in silos. So medical imaging data is stored in as DICOM files in PACS systems. And these PACS systems work very well if you have a clinical setting and you just want to be able to look at the images to make your radiology reports. But these PACS systems are not really developed to share data, or they're not developed to search through, to query your data to develop your AI algorithms. So data is in silos, and that results into limited access to medical images. And also sharing data between imaging centers is really hard because the data is stored safely in these PACS systems. Small sample sizes and a lack of diverse geographic areas hinder the generalizability and accuracy of developed models. So for example, you can develop an AI algorithm on a specific population, which is what usually happens. You have one hospital or maybe two hospitals populations that you use for your AI development. And then if you would validate and test it in this same population, it will probably work pretty well. But if you would try it out in another population, another geographic location, and maybe another ethnical or age distribution, then it might not work at all. So bias is a very strong, very big problem right now in AI, because you need heterogeneous data to be able to develop unbiased or relatively unbiased algorithms. Here you can see an example which I came across pretty recently. Here, there are 3 models that look at COVID-19. This is training data. You can see that the confidence of these models is really good. Then you have unseen data. You can see that the confidence goes down. And then randomly, they just showed a photo of a cat, totally unrelated data. And for some of the algorithms, actually, the confidence was really, really high. Well, this obviously has nothing to do with the chest X-ray. So this bias is a very big problem in the medical imaging space right now. So all those small data sets, they may be sufficient for training of AI algorithms in a research setting. Large data sets with high quality images and with annotations are still essential for supervised training and for validation and for testing, mostly of commercial AI algorithms. So small sample sizes is also still a problem. Other problems are that data needs to be anonymized to be able to work with it. You need high quality data. The data needs to be structured or organized. And oftentimes, if you want to develop supervised algorithms, you'll need data to be labeled. So in order to tackle all these problems, there is a pathway that leads to high quality data. And during this presentation, I will go through all these steps and we can see what is needed to prepare your data. And I'm mostly doing this from a perspective of a radiologist or a resident that works in a hospital and that wants to develop his or her own algorithm or wants to try to play with it. So the first step before you get access to your unprocessed image data is to get ethical approval. How do you get ethical approval? Well, you need to write a proposal for the Institutional Review Board or the IRB. And there are two options. You either do a prospective study or you do a retrospective study. Prospective studies have higher quality, they're more expensive, and yeah, they're generally just harder to do because you still need to get all the data. But the biggest advantage is that you can choose what kind of data you want to collect. So the quality, therefore, is usually much higher with prospective studies compared to retrospective studies. Retrospective studies are way more common. Those are the studies based on data that's already present. And this is the currently the bread for machine learning algorithms because the data is already there. So it's fairly easy to do it. It's also but the disadvantage is you have to deal with the data that is already collected. So you cannot choose any other data that you want to use. You just have to deal with whatever is there. Another disadvantage of prospective studies is that you will need active informed consent from all the participants of your study. And with retrospective studies, the IRB will often waive the need for informed consent. So there are some pros and cons, but retrospective studies are currently the most commonly used study design types in machine learning algorithms. But either way, if you go for a prospective study or a retrospective study, you will need IRB approval. So once you've gathered your IRB approval, you can start to get access to the unprocessed image data stored in the PACS. So generally spoken, medical imaging experts, professionals have access to PACS systems. For example, physicians, technologists, PACS managers, and clinical scientists have access to PACS. But these are usually not the people that develop the AI algorithms, because the AI developers are typically not located within the hospital, and therefore they often do not have direct access to PACS. So ideally, there would be a collaboration between physicians and AI developers. And this is mostly to develop an algorithm that's really, that results in a research project that can be published, or that's going to result in a commercially available algorithm. If you want to play around with the data yourself, of course, that's not really relevant. But still is an important point, I think. Okay, so once you have access to the data, you can start querying the data. So what this means is you only want the data that you're going to need for your specific algorithm. So you need to be able to search to find the customized data that you're gonna need. So once the data are available to the AI developers, there are different strategies to search for the medical images and for the clinical data, if you also want clinical data next to your medical images. So search strategies, they may consist of strings or words that you can search for in radiology reports, ICD-10 codes, or current procedural terminology codes, for example. And searching, the searching of these strings can often be done by searching through PACS and electronic medical records by using search engines provided by PACS or RIS systems. And if your PACS system or RIS system does not have such a search engine, there are alternatives. You can get software packages that simplify the whole process of the data querying. There are commercial options such as Nuance and Eliminate, and there's also non-commercial options. They're not very common, but within Stanford we have the STAR system. So STAR is Stanford Research Repository Tool, and it's pretty unique to Stanford. There are not many hospitals or universities that have such a system, so that is unfortunate, but it's good for us that we can use this. And maybe there will be developed more of these kind of tools will be developed at other hospitals. So with the STAR tool, there are 3 steps to be taken before you can get full access to your data. First, you can do cohort discovery. So you can build a cohort using demographics and clinical information from the electronic health record. So this means, for example, you can say, okay, I want to develop an algorithm based on adult patients, so 18 years and older, and they have to be females, and they have to have aortic dissection or whatever disease you want to evaluate. So that's how you can discover which cohort you want to use. The next step is the compliance step. So based on the search that you've done in the first step, you can get some basic statistics of the selected cohorts. For example, you get like the mean age, or you get the ratio male-female, or you get what kind of ethnicities are present. But you don't get an individual information about every patient. But because before you actually get full access to the data, our university requires that you show that you have an IRB approval or data privacy attestation. So once that is provided, then you can do the actual chart review and you can download your data and you get full access to the data. So this is how the STAR system works. And it actually works pretty well. The disadvantage is it's mostly focused on clinical data and not on medical imaging data. So it's not very applicable to radiological research projects, except for we use it to get clinical information about the cohort that we already selected based on the imaging data. So once we have selected the patients that we want to evaluate in our study, so once we have queried our data, we're going to go to the next step, and that is to de-identify the data that's present. So either if you are in the US and you're following the HIPAA guidelines, or if you're in Europe and you're following the GDPR guidelines, it is very essential to de-identify your medical data before you use it for AI development. This table shows the 18 identifiers according to HIPAA that need to be removed from your data. And identifiers include, for example, obvious ones such as the name of a patient or the date of birth of a patient, but also medical record numbers need to be removed. And there are more identifiers than you might think there are. So it's very important to remove these identifiers from the data. So medical imaging data is stored in DICOM files. So DICOM files consist of two elements. One element is the pixel element. So these are just the images that we are looking at. And the other element is the metadata element. So these are the DICOM headers or the DICOM tags. These DICOM headers, they contain information, for example, the patient name and the patient medical record number and date of birth and these kind of things. So there is definitely PHI or protected health information present in these DICOM headers. So it's important to remove those. Luckily, it's relatively easy to remove PHI from DICOM headers. One of the easiest steps is just to convert your DICOM file to an only pixel or voxel based image, for example, to a PNG file, or you can convert it to a NIFTY file. This way your whole DICOM header will be removed, and you'll also remove the identifiable information. The disadvantage is you also lose a lot of relevant information that might help your algorithm, because you, for example, also lose what is the age of this patient or what is the gender of this patient, that might be relevant for training your AI algorithm. So you got to be careful with that. There are also other de-identifiers available. If you just Google DICOM de-identifier, you'll find some open source options, and also many PACS systems or image viewing software has de-identifiers built in, which you can use, especially if you do like a small project within your own hospital, and you can do them one by one. You don't have too large of a population. That could be an option. A more difficult problem is to remove PHI from embedded or burnt into images. Especially if you want to do something with ultrasound or echocardiography, PHI is often burnt into the image. So the name of the patient and the date of birth, for example, is often on the actual images. And in order to remove that, you need to, for example, put a black box on top of it to make sure that these pixels do not show this PHI anymore. So this is a little bit harder to do. Most imaging modalities don't have this problem, but some of them do. So this is something to take into consideration. And the hardest problem is if the images themselves actually contain PHI. So here you can see an example. I downloaded a few head CT scans from the RSNA Kaggle challenge, and I created some 3D reconstructions. And here you can see a patient that got a trauma head CT scan. And if you do a 3D reconstruction, you can actually see the facial structures of this patient. And that is just a way to re-identify specific patients. So you got to be very careful with these kind of things. And this is obviously mostly a problem if you are dealing with body parts, such as the head. Like if you are developing an algorithm for coronary CT angiography, you don't have to worry about it because it's hard to recognize somebody based on the coronary anatomy. But for certain body parts, this is a problem. Okay, so once you have de-identified your data, the next step is to transfer your data to a local or to an external storage. Local storage is often used for single center studies, and external storage is used for multi-center studies or for commercial AI development. So the pros of local storage are data remains within the institution, so it's relatively safe, and also it's relatively available. And the pros for external storage are that sharing data is relatively easy, and especially if you use a cloud-based external storage method, backing up data is fairly easy. The cons for local storage are that data sharing is really hard because it just remains within the institution. And the cons for external storage, especially if you use cloud-based, are high costs and the need for a fast internet connection because you're dealing with large amounts of data. So once you've transferred your data to a local or an external storage, you need to control the quality of the data that you're dealing with, because the problem with a lot of data that's extracted from PACS systems is that it's very chaotic. One of the things, for example, that you could control the quality is the type of the exam that you are looking at. For example, if you want to develop an algorithm that works with chest x-rays, you can extract x-rays from the PACS system. And this is an example of what you could get. This is actually a real life example from our startup. And you see all these exams were marked as chest x-rays, but there actually are some exams that are not chest x-rays. So you need to control what exam types you are dealing with and if they are indeed what you, because you will see there's always some outlier. So what we do is we create a large matrix and we can see many exams at once, and we can select the ones that are different. And if you do a small study within your own hospital, then you can just go by every patient and just see what kind of images you're dealing with. And if there's one patient with the wrong exam, you can just simply exclude these. Another thing that you want to look at, for example, is image quality. If you want to develop algorithm on cardiac CT and there is breathing artifacts, or a patient had an extra systole and there's a cardiac motion, then you want to know this. Either you want to develop an algorithm that only works with high quality data, or you want to develop an algorithm that works on all types of cardiac CT scans, also low quality, but then you want to know whether the image quality is low or not. So this is another feature that you can do quality checks on and make notes. So after you've learned more about what the quality of your data is and maybe excluded some of the low quality exams, it's important to structure your data. As I said, the data that you extract from PACS systems is often very chaotic. For example, and this is just one small example, but the DICOM headers might be totally different in different exams while they actually have the same protocol. So if you use a different CT vendor or the CT scanner, there are two CT scanners. One is located in the emergency department and one is located in the outpatient department. And they might have the same exam, for example, a coronary CT angiography, but the protocol name mentioned in the DICOM header could be CCTA or coronary CTA or coronary CT angiography. Well, we all know that this is actually the same protocol. The the names might be different. So this all needs to be structured. Another example is if you want to work with cardiac CT, is you want to know whether these exams are ECG synchronized. So first of all, you want to know yes or no, and if so, is it prospectively ECG triggered or is it retrospectively ECG gated? So there's a lot of information that you need to structure to make sure that your final algorithm knows what's going on in each exam. So once you've structured your data, we get to the last step, and that's to label your data. And there are different types of labels with that add different types, value to your algorithm. The most easy to get label is retrospective annotations. This, for example, could be you have a chest X-ray and you see an atypical lesion in the left lung, and you can mark that. The disadvantage is it's retrospective data, so you cannot really look into the data anymore. What is the history of the patient? Was this a malignancy? Was it maybe an infection? What was the cause of this? It remains unclear because you don't have much information. So more value is provided by prospective annotations. And this is, for example, prospectively gathered information. So you can still look at clinical information such as the electronic medical records, or you can look at lab results, or you can look at pathology or the history of the patient. So the value of these kind of annotations is much better. But the most value you get from the actual ground truth. So for example, if you want to develop an algorithm that automatically detects lung nodules on chest CT scans and that determines whether these nodules are malignant or benign, you would want to have the ground truth and actually know if these nodules are malignant or benign. So if you have biopsy information, or if you have follow-up and you know that this patient died from lung cancer 6 months later, then you know that this was actually a malignant lung nodule. So the ground truth is the highest value, adds the highest value to your algorithm, but it's also the hardest to get. Here we can see an example of a chest CT scan with a solid lung nodule, and then I'll show you some different types of labels. So for example, you can have a dichotomous categorization or classification label. For example, lung nodules present or absent. Or you can have a quantifying label such as lung nodule, the maximum diameter is 4 millimeters. Or you can have a multi-class classification. For example, this lung nodule is benign, malignant, or unknown. So these are all different types of labels that can be either on the level of each image, each series, each exam, or on a patient level. Another type of label is a segmentation, in which you get the coordinates of the specific lesion that you are looking at. It can also be relevant. So how do you get these kind of labels? Well, there are different platforms available. You can do it if you have your own, if your hospital has their own software package, you can use that. Or for example, Daniel Rubin here at Stanford developed ePad, which is an open access, open source platform. You can just use it in your web browser and you can do segmentations or labeling there. But there are many other options you can use to label your data. And then who is doing the actual labeling? Well, there are kind of two options. You can ask a crowd, so use crowdsourcing. These are non-experts, but many people. Or you can ask a small number of people that are experts in the space. And the experts obviously is used the most, but there actually are some examples of crowdsourcing. For example, Haim et al. Published in Journal of Medical Imaging in 2018, a study in which they had the crowd segment organs, under which the liver. And then after the segmentations, they used majority voting. So they asked the crowd again, which segmentation do you think is good? And then they came to a final segmentation. And they actually found that crowdsourcing can be used to create organ segmentations with similar quality to those created by single medical expert annotators. And they said that engineers with domain knowledge and medical students and radiologists were medical expert annotators. And I think that this might indeed work. Simple tasks such as segmentation of organs that are easy to delineate, such as the liver. But I think it gets harder if you want to classify specific, maybe more rare pathology on imaging exams. So I think it will not work always, but for certain tasks, maybe the crowd could help with labeling images. Also, a lot of labels are extracted from radiology reports. It would be much easier if we would have structured reporting. Unfortunately, that's usually not the case. We don't have that. The reality is we often have relatively unstructured reports. And what is often used is NLP algorithms to extract the information from those reports. So going through all these steps, as you can imagine, it's quite a pain. It's quite a bit of work. And if you just want to get your feet wet and try some things out yourself, you can also just download publicly available data. The Cancer Imaging Archive and the RSNA Kaggle challenges, for example, and also within Stanford, we have some data sets that are freely available that you can use for your AI algorithm development. Here you can see a table that we published in the paper that Merel mentioned before in radiology. That shows some public available data sets. So the disadvantage of these ones is that the number of labels and the quality of the labels is often somewhat limited. So if you want to develop something substantial, something that's going to be used in the clinics, then this is not going to be enough. But if you just want to play around with it, it could be very interesting. I also wanted to tell you something about the process of medical imaging data preparation that might change in the future. So currently, we are using centralized AI model development, but maybe in the future, we might go towards federated or distributed learning. So what currently happens if you do a multi-center study, for example, with 6 hospitals, you send the data from each hospital to a centralized data storage, and that's where you develop your algorithm. The disadvantage is you have to de-identify your data, and you have the risk of data breach. The federated or distributed learning development is different. You don't send data from a hospital to centralized data storage, but you send a model to each hospital. So what happens is you start with the basic model, you send it to a first hospital, you train the model based on the data that's present, and instead of sending the data back, you only send the updated parameters of the model back. And then you go to the next hospital, and so on and so on. So this is the sequential way of doing federated learning. There's also a parallel way, and there's different approaches on how to do federated or distributed AI learning. There are some major problems because labeling images within each silo is going to be really hard. Organizing, structuring the data, it's really hard to do that in a similar way. So there are still some technical problems, that challenges that need to be tackled before federated or distributed learning is gonna be applied. But I just wanted to mention that this is something that might be in the future horizon. Then I want to shortly say something about SegMed, the startup where I'm a co-founder in. So our mission is to empower medical AI development, and we want to help patients and clinicians all over the world to have access to high quality healthcare. And how are we going to reach this mission? Well, we are currently building an indexed search library that is curated specific for AI development. So the data is easily ingested by developers, and developers can customize their data sets very easily. So the whole data pipeline that we talked about before, with de-identification and standardization and labeling, it's all provided within the SegMed cloud. And additionally, there's a search method to actually search the data that you want to do, and you can create your own customizable data set. So how does it work? SegMed is collaborating with hospitals all over the world. The hospitals upload their de-identified medical data to the cloud. It's only uploaded after it's checked for potential PHI. So only fully de-identified data can be uploaded to the cloud. Then the cloud makes sure there is a diversity distribution because there are collaborations with hospitals all over the world, and not only, for example, one place in the United States, but multiple states, multiple areas. So you really get heterogeneous data, so you can reduce the chance of having biased algorithms. Then the data is all standardized in the same way, and if needed, the images are also annotated. And then the last step is that the AI developers, they can use these data sets for training, validation, and testing purposes. I'm lucky to have a a great team. Kaylin is our CEO. She worked at Apple Health Tech and Verily Life Sciences. Adam has more than 10 years of experience and is our CTO. He works in he has more than 10 years of experience in the Silicon Valley. Jay is our AI director. He has worked at Facebook and is currently finishing his PhD at Stanford. We also have some great advisors, such as Matt Lundgren from the Stanford AME Center, Hugh Harvey, Tim Leiner, and some people from industry, such as Julian Wichmann. If you want to read more about our startup, you can look at segmed.ai. And if you want to read more about the whole process of preparing medical imaging data for machine learning, I recommend reading the paper that we published April this year, together with my co-founders from SegMed and with our advisors and friends. We published this paper, and it gives a more in-depth view of what I have just been presenting. So I want to end with acknowledging these people that have substantially contributed to the information that I was presenting to you today. So thank you for your attention, and I'm more than willing to take some questions.
All right, thank you very much, Martin, for this really excellent presentation. It really gives a nice overview of what radiologists or other specialists should do when willing to start training an algorithm themselves, and how to care for the data or how to prepare the data. So this is really essential information. There's quite some questions, so I would like to give the word to Merel to ask, maybe to launch the first question.
Yes, thank you, Eric. Well, the first question, I just answered, typing the answer, but I just want to point out that all our webinars are available at our YouTube channel. So if you missed something or you want to take notes of a certain slide, please go to our You Saw Me YouTube channel. So the first question, we have quite a few, but we still have about 15 minutes. The first question posed by Anonymous: what's the scope for services startups doing a curation, annotation, or labeling for developing medical AI? So do you think there's a demand? So actually, I think this is the ideal question for you, Martin, being co-founder of SegMed.
Yes, yes, it's a very good question. There is a very high demand. So every startup or every service, every medical device company that develops AI algorithms needs high quality data. They need curated, annotated, and labeled data. So yeah, the demand is tremendous. There's definitely a high demand, because if you don't have this, you cannot develop quality algorithms. And like I said before, if you want to develop something yourself as a radiologist and want to with it, it's fine. You can just, you don't need necessarily high quality data. But if you want to develop something that's going to be FDA approved, you really want high quality data. And that's what all these startups and companies are also realizing. So yes, there's a high demand for that.
Okay, thank you for answering this question. There's two questions related to the GDPR. Of course, you're located in the United States, but you also know about the GDPR without doubt. So the first question is, well, first of all, the attendee is Sanam Asili. Thanks you for the great presentation, but he wants to know how we can deal with the GDPR and what steps need to be done. And then the second question is also asking about GDPR. It actually does not require pseudonymization. So do you have experience with that? Could you recommend pseudonymization, and maybe what's the main difference between both?
Yeah, okay, let me start with the pseudonymization question. So the difference between anonymization and pseudonymization is pseudonymization means that there is some sort of code that you can still retrace which patient you were dealing with. So for example, you have a secure Excel file somewhere that says the patient with this ID equals the patient with this medical record number. And generally the researcher does not have access to this, but maybe the original data provider has access to these codes. So full identification is more thorough than pseudonymization. The advantage of pseudonymization is that retrospectively, you could theoretically still get more information. Let's say you develop an algorithm for lung nodules, and later on you're working on this for a year, and you think, hey, maybe in the last year something has happened. I would like to know if something happened to these patients. Then you can go back to the data partner and say, hey, I would like to know for these patients what happened over the last year. Can you look it up? And then you can add information to your data, and that's not possible if you fully de-identify. Yeah, GDPR indeed says pseudonymization is enough, while de-identification is more thorough than pseudonymization. So if you can use pseudonymization, yes, I would definitely recommend it. But if you want to be very, very thorough, and you want to be very, very sure that you won't that you won't get any identifiable information on the streets, then I would recommend to go for full de-identification. Regarding the other question, what needs to be done to deal with GDPR? Yeah, so
that's it.
Sorry, Eric, what did you say?
Yes, that's the question. What steps need to be done? Yes.
Yeah, so first of all, you need to really thoroughly go through your data and see if there's any, I mean, the most important thing is to make sure that your data is de-identified. If your data is fully de-identified, then you are complying with GDPR and HIPAA. And then you can even export your data to other hospitals or to other researchers or to other continents, as long as it's fully de-identified. Because if it's fully de-identified, it's not considered patient data anymore. So that's, I think, is an important factor to realize. And what tools can be used? Yeah, so there are, like I said before, if you want to de-identify your data, there are quite some tools available. That's actually not too hard. I know, for example, the example that I showed when you can make a 3D reconstruction and you can get identifiable information based on the image. There are tools available for brain MRI scans that scramble the surface of the MRI scan. And if you make a 3D reconstruction, you do not recognize the facial structures anymore. So there are definitely tools available. I recommend reading our paper where we mention some tools, or you can also just Google and search around a little bit if you want something specific.
Yes, maybe one important thing to add is the fact that you need a real purpose to use data from patients. So you have to, certainly if you go for research, you really have to define the purpose of using the data. But of course, you're right, if you fully anonymize the data, then the GDPR is not applicable.
Yeah, exactly.
All right, so the next question was posed by Michael Desert. It's about quality control. So you use the DICOM format. He says, if I'm correct, can it include lossy JPEG images with artifacts or lossless images, so being raw images, depending on the imaging system? How do you homogenize data? How do you filter them per system and per high or low quality? That's a complicated question. Do you know the answer to that, Martin?
Yeah, those are a lot of questions
yes.
in once. Yeah, so I know, I mean, you can use different file types, JPEG, PNG, TIFF, and with different types of quality. So obviously, if you have JPEGs with artifacts, you will work differently than if you have images without artifacts. Yeah, I don't know how to answer the other questions.
Well, that's okay. We have a lot more to go. So
okay.
Let me jump to the next question then, by Oliver Diaz. So high quality images are desired, but shouldn't AI also learn from low resolution images? for example, fast sequences. So there's no point to train with idealized images if they are then not available in the clinical routine. Which I actually quite like this question. This is also some form of bias actually we're referring to here.
Yeah, no, I totally agree, and it's a very good point. So actually, what I mean with high quality data is that you actually know what's going on in your data. So I think if a heterogeneous data set includes also low quality data, low image quality, but you have to know that your images have low image quality. If you don't know whether all the images are good quality or low quality, then you don't really know what you are developing. So I think it's important to do your quality control and then decide, what do you want to do? Do you want your algorithm to work on just perfect images, or do you want to your algorithm to work on every daily routine clinical image? So I think it's a very good point, but you will still need to do your quality control and then decide what quality you accept within your data set.
Great. So we align the data set with the real world data, so to say.
Yeah.
Cool. So the next question, this contains a very nice word, I think. So Nicolas Papanikolaou asks if Stanford has produced some super popular cyber radiologists. Yes, actually, it's true. So they're meaning he means that they are familiar with the technical aspects of AI. Do you think that this should be the way to increase AI penetration to clinical practice in other places? So are these people examples, or can we also do it another way?
Yeah, so I mean, these people are people that understand both the technical side and the clinical side, and that is definitely the way to go. If you are an AI developer and you don't have any clinical or medical knowledge, it is very important to partner either with a radiologist or a clinician to make sure that what you're developing is actually, that there's actually demand in a clinical setting, that it's actually clinically relevant. And the advantage of these cyber radiologists obviously is they know what's going on in the clinics and they know what's going on technically. So yeah, that's totally the way to go.
Okay, we have more questions to go now. So there's a question from Maurice Panner. He's asking actually if there's any accessible Python libraries that can help with some of the steps you mentioned.
There probably are. I am not familiar with it, but you would have to ask my CTO or technical co-founders, and they will definitely have an answer to this question.
Okay, so let's go to the next question then. There's Tiago Lima asking, so again, you mentioned about the data quality. So one challenge with MRI datasets is the heterogeneity of images. So what are the steps you recommend for working with such data?
Yeah, it is totally true. So what is important, again, that you know where the heterogeneity is, how the MRI images look, and what you want to use for your algorithm. Maybe because the heterogeneity is in that you have so many different sequences, you only want to develop an algorithm that works on specific sequences, so you can select those ones that are relevant for your algorithm, and you can just disregard the other ones that you maybe don't need. Yeah, so you have to balance between genius do you want your data to be and how functional is your algorithm going to be in the end.
Okay, thank you for that answer. We will have to make a selection of questions because there's too many, I'm afraid. There's an interesting one. Will we have artificial intelligence doing all the labeling preparation at high quality anytime? So what do you think about that? Or will there always be some human in the middle to make the selections and the preparations?
Well, obviously, like within our startup, we're trying to automate the process as much as possible. We're using a lot of NLP. We're using methods to make sure that we need as least radiologists as possible. But what we are experiencing is we do need radiologists and experts that actually have experience with looking at these images to label the images. So I definitely think that a large part of the process is going to be automated, but I also think that there will always be a need for people that understand the clinical or medical part of this, that can make sure that the automatic methods are guided towards proper clinical applications.
Yes, and I know those tools, they already exist, for example, also for labeling or segmenting images. So you have semi-automated tools that help you finding the right lesions, etc., automatically, and that train themselves as well. So there will be improvements in the near future, that's for sure. There's also a good question from Sotiris Bizdas. How can you effectively make the most out of the EHR to curate and label your data? Are there any pipelines available?
Yeah, so like within Stanford, we have the STAR system. So that's a pipeline that is available. There are some, I know that in the UMC Utrecht, for example, Wouter Veldhuis is developing a method to organize the data that's present in hospitals, and that might be helpful. But so far, this is something that still needs to be developed in a lot of places. So if you have an EHR where impossible to query data, yeah, you might have to go to a commercial solution.
Yes, but actually what you would need is like some platform that you can attach to your PAC system to do this, to make this kind of semi-automated. And I think what you're referring to, what Wouter Veldhuis developed, is the Imager platform. But this will be commercialized by Sectra now. So, but yes, I think this is
so it's another commercial solution. Yeah, it
is. Okay. So to increase, there's another question about the data availability. So can we create our own data if you have if there's not enough data available?
You mean synthetic data?
Yes.
Yes, so there are methods to create or generate synthetic data. As far as I know, the mostly used technique for this are GANs, generalizable adversarial networks, and they seem to work pretty well on normal data, on normal images. So as long as there is no pathology present, it is kind of possible. But if there is any pathology present on the images, then it gets more problematic. So I'm sure these kind of techniques will be developed better in the future, and this will be available. But currently, I don't think, at least as far as I know, we have techniques available to generate really high quality synthetic data.
Yeah, I agree. I just typed an answer. I think that's a good answer, Martin.
Okay, thanks.
Okay, I think we can stop the webinar at this moment. So thank you very much for having given this excellent lecture. And actually, I also would like to motivate the attendees to read the paper that you published, because that's a real good paper. And I also know that it's very practical and applicable. Yes, that's the one. Thank you for showing. Okay, so I would also like to thank all the attendees for having been here. Of course, all the panelists. And Merel, thank you very much for having done this. And of course, we also have to thank our sponsor, Bracco. Don't forget, you will be able to look again, or you can even forward the this webinar if you want to colleagues or within your network. The webinar will be available on our Usomi website. And yes, we would also like to encourage you to become member of our society, because we want to integrate knowledge of different experts, not only radiologists, but also the AI developers. We want to close the gap, which is also, I think, quite a crucial issue, as Martin also mentioned in the beginning of his presentation. So we should really enhance the collaboration between the computer scientists and the medical doctors or the radiologists. So again, thank you very much, everybody, and hope to see you for our next webinar. Have a good evening.
Bye-bye.
Thank you.